el apoyo muy generoso de un programa de Naciones Unidas que se llama UNU Biolac, de FOSEM y de la Embajada de Francia. Y sin duda esto tampoco hubiese sido posible si no fuera por el apoyo logístico del Instituto Pasteur, en particular de Virginia Ron, la Secretaría, el equipo de Secretaría General, Natalia Luján, Mariela y varias personas más que han estado estos 10 meses atrás de toda la gestión. Del workshop están participando 24 estudiantes de Argentina, Brasil, Ecuador, Perú, Chile, México y Uruguay. Tuvimos la suerte además de contar con, además de las personas que ya nombré, la participación de profes que vienen de otras partes del mundo. Cristina Landeta, de Indiana, Estados Unidos, Begoña, de Australia, de Trove University, Marco Mariotti, que lo perdí de vista, allá en la esquina, de la Universidad de Barcelona, y Memo Bergman, de New England Biolabs. Lo que sigue es el final de este workshop, que ya sucedió los primeros 2 días de esta semana. Quiero agradecer muchísimo a los y a las participantes, a todos los que hicieron posible, a todos ustedes por estar acá, y a los estudiantes y a los estudiantes que metieron muchísimas ganas estos 2 días. Todos participaron, todos se presentaron y fue un esfuerzo para ustedes. ¿Ok? Voy a pasar a inglés ahora para presentar a Jean-Paul. It's very funny that you have this.
You make me wear that.
No, so the first speaker from this morning is Jean-François Collet, is professor of medicine at the University of Louvain, Belgium, and also the head of the Bacterial Stress Response Lab at the Duke Institute. Short introduction, as I say, nowadays all the CVs are available online, so you can search for the information of the people if you want to know more. Jean-François has been working in the redox microbiology field for more than 20 years. Key publication for all of us. The XB system first, and then moving to something more related to stress in the periplasm, and also discover many of the enzymes that have been studied for the last few years. So it's a pleasure to have you here. Thank you very much, and go ahead.
Thank you. Thank you, Bruno. Thank you, Memo. Thank you all for the invitation. Thank you for being here or joining online. So it's my first time in Uruguay, first time in South America. So it's been awesome, and I'm very, very glad to be here. So in my lab, I think that the common question that people ask is, how do bacteria do to face this? I mean, to face stress, and so as you know, stress can come in different flavors. And one type of stress that we study is osmotic stress. And this morning, the first story I'm going to tell you, because I'm going to tell you 2 stories this morning. So the first story I'm going to tell you is about osmotic stress. So it's not related to redox, but so together we're going to ask that question. How do bacteria do, how gram-negative bacteria do when they are exposed to a sudden drop in osmotic pressure? So that's why one kind of stress and why one question we ask in the lab. Another question we ask is, how do bacteria do to deal with oxidative stress? And this morning, I'm not going to talk about that. For those of you who are going to Punta del Este, I'm going to give a talk there on that topic. The third question that many people in the lab ask is, okay, how do bacteria do, or how do gram-negative bacteria do to protect their cell envelope? The outer membrane, peptidoglycan, how do they do to protect those layers from stress, to feel stress? And that's the second story I'm going to tell you this morning, okay? So first, it's really like my talk is made of two different stories. And so after the first one, if Bruno is okay with that, I can stop for a few minutes. And if you have questions on that first story, and I think that probably that will be the best moment to ask them. And then I will proceed and go on with the second story. Okay, so let's start very simple. So that's a bacterial cell, a gram-negative. Could be E. coli, could be Salmonella, whatever. And as you know, bacteria, in the cytoplasm, in the periplasm, they have a very high concentration of macromolecules. It's been estimated to be like 300 milligrams per mil. And because of that, so they face a difference in osmotic pressure with the environment. In fact, they maintain that they need to keep an internal osmotic pressure that is higher than that of the environment. And this is required for them in order to grow and divide. So, as you know, bacteria live a very versatile lifestyle. So in a fraction of a second, they can find themselves in a completely different environment. Like for instance, they could go from LB to water, or they could go from one medium to another one, to a lake or to a river or something, where the osmotic pressure will be much lower because there are way less osmolytes. And so what will happen is that, when the osmotic pressure drops, then water molecules, in blue there, will enter the bacteria, and this will lead to an increase in the internal osmotic pressure. This will push on the membrane, and so the volume will expand. And if bacteria didn't have something to protect them from that increase in internal osmotic pressure from turgor, they would lyse, they would explode, and we wouldn't have a job, and in fact, we would not even be here on this planet, okay? So we know that it's not the case. So we know that there is something, that allows bacteria to resist those sudden drops in osmotic pressure. And if I ask you, what's that thing? What will you tell me? What prevents bacteria from lysing when there is a sudden drop in the environment in the osmotic pressure of the environment? Come on. Sorry? Pumps? No, there is something like a device, something like that will prevent. No? Peptidoglycan. Thank you. The cell wall. So in fact, if you go online and if you look for reviews on the cell envelope, on the peptidoglycan, on osmotic stress, these, are the kind of sentences, paragraphs that you're going to find. And these are from friends like Valdemar, in Australia now, like he's like a world expert on the peptidoglycan. And this is what he wrote. Most bacteria surround their cytoplasmic membrane with a stress-bearing peptidoglycan layer, also called sacculus, that protects cells from bursting due to its turgor. And the two other paragraphs, they basically say the same. Like here, so it will generate osmotic pressure, it pushes the plasma membrane against the cell wall. The cell wall defines the cell shape and constrains the volume and the turgor. So that's something that you find in all textbooks, is that the cell wall will protect bacteria from lysing, when there is a drop in the osmotic pressure of the environment. And so in the first part of my talk this morning, I'm going to show you that this is not entirely true. In fact, the peptidoglycan is necessary, of course, but it's not sufficient. So it's not the only answer. On its own, the cell wall does not protect bacteria from lysis. So this story starts with Seung-Young. Memo knows Seung-Young, they were together in the lab of John Beckwith, at Harvard. And so Seung-Young has been working with me for 13 years, and so he's working on the assembly of the cell envelope. And, as he was working on one of his favorite projects, he made the following observation. He found that when you have a double mutant that doesn't have LPP and OMP, I will tell you in a moment what those two proteins do, okay? So, but just keep in mind that he found that when you don't have, when E. coli, we mostly work on E. coli, when E. coli doesn't have LPP and OMP, then when you take this double mutant and you shift the cell from LB to water, then, as you see, they don't survive. They become extremely sensitive. So now I'm going to tell you what those two proteins do. So LPP is a lipoprotein, so it is anchored in the outer membrane with a lipid moiety, okay? And then it is covalently attached to the peptidoglycan. So it's a bridge, it's a tether from the outer membrane to the peptidoglycan. The other protein is OMP, and so OMP, here, yeah. So OMP is a beta-barrel protein. I mean, it's very abundant, like LPP is. LPP is, like there are 1 million copies of LPP in a single cell. So it's the most abundant protein in E. coli. OMP is also very abundant, like 200,000 copies. It's, of course, it's a beta barrel, so it's in the outer membrane. And as you see, like there is a delay. So there is a periplasmic domain, that will bind to the peptidoglycan. So it's also a tetra. It also connects the outer membrane to the peptidoglycan. Okay, then Sung Young said, okay, but those two proteins are very abundant. Maybe that when we delete them, when we delete the corresponding genes, maybe that we change the properties of the outer membrane, maybe that we change the proteome so much that the cells just become more fragile. And so to know whether it was the case, what he did was to engineer two mutations in OMP and in LPP. So you see here the LPP delta K58. So it's the last residue of LPP which is deleted. And when you delete that residue, you cannot bind LPP to the peptidoglycan, but you keep the protein. You still have the 1 million copies in the outer membrane. And in the other one with OMP, you change, an arginine by a glutamate. It changes the charge, and OMP doesn't bind to the peptidoglycan anymore. Okay, but we have the two proteins there. And as you see, when we specifically prevent the attachment of those two proteins to the peptidoglycan, we have the same phenotype. So it's not the absence of those two proteins that is the problem, is the fact that the outer membrane is not attached to the peptidoglycan. And that was extremely surprising, because remember those reviews, that I highlighted, remember this idea that it's the peptidoglycan that protects the cells from bursting, from lysing. Then why preventing the attachment of the outer membrane to the peptidoglycan? Why? Why does that cause this sensitivity? Why do cells that do not have an attached outer membrane, why do they die when they are exposed, to a drop in the osmotic pressure of the environment? I mean, the current model did not explain that. And so we decided to investigate that further. And that part of the work was done by another senior scientist in the lab, Michael de Gelt, and he likes to use microscopy. And so he decided to use, single cell analysis. So what Mike did, was to do the same. So to take E. coli cells, wild type, mutant cells, to shift them from LB to water, but under the microscope, and to look at them at the single cell level to understand what was going on. And, if you want to do these experiments, and if you want to extract information, if you want to dissect, how cells die, then you need markers. And we decided to use 4 different markers. First, we decided to label the two membranes, the inner membrane and the outer membrane, using those two fluorescent protein. So LPUB, it's an outer membrane lipoprotein fused to M-cherry, and then GLPT, it's an inner membrane protein fused to GFP. And then we had a second set of markers, and here we simply express M-cherry in the cytoplasm, soluble M-cherry, or turquoise in the periplasm. Okay, so now we have those 4 tools, and we're gonna look at how the wild type and the mutants behave when we shift those cells from LB to water. This is unpublished. Okay, so this first story is completely unpublished. If you are interested, you can go on bioRxiv and you look for Michael's name and or my name, and you will find the preprint. Okay, so this is what we could see when we did this analysis with the wild type. Basically nothing. So there is a movie there. You see it changes, it moves a little bit, but basically you don't see much. So when we take the wild type, and we take it from LB to water, there is not much to see. And that's not surprising, because on the spot titer assay, we could see that the wild type, could survive this osmotic stress. When we, look at snapshots, again, they don't change. The markers are not impacted by the osmotic shock. The same is true when we use the other set of markers. So M-cherry in the cytoplasm and turquoise in the periplasm. Again, okay, nothing is going on when we look at the wild type. Things are going to be very different when we do this experiment with the mutant. Okay, so remember the mutant, here we don't have the other membrane attached to the peptidoglycan. So when we use the two membrane markers, look at the movie on the right, you're going to see lots of things going on, and then I will go slowly through the different steps. See, so boom, there is like this bleb forming, this bulge, it relocates, and then the red color is lost. So I will play it again, and you see, yeah, so obviously something is different when you do that with the detached outer membrane. And again, something very similar when we use, the soluble markers. Okay, I'm going to start the movie now. Okay, you see this bleb forming, then the blue color is lost, and then everything is gone. Okay, so it's very complicated. So we spend hours looking at those movies. And so now what I'm going to do for you, to make it a little bit more simple, we are going to focus on 3 key events. Okay, so the first one, when we look at the outer membrane in blue, we see that after a few seconds, so this is 5 seconds after putting the cells in water, we see this bleb forming. Okay, that's the first thing we see. And then second, when we look at the periplasmic signal, so M turquoise in the periplasm, we see that more or less at the same time, all the periplasm relocates to that bleb, the second thing, and then it's gone. You see the blue signal is gone. And then the third thing is when we look at M cherry in the cytoplasm, we see that, here, it will move, in fact, to the periplasm, and then it's gone. Okay, so the 3 key events to keep in mind, because then we're going to try to build a model together and explain what we are seeing. So 1, bleb formation. Second, accumulation of periplasmic content in the bleb, and then, the periplasmic content is released. And then 3, the cytoplasmic content is transferred to the periplasm and then released. Okay. So what do these data tell us? Okay, so before the shock. Okay, so now we're going to zoom at the cell envelope. So you recognize the 2 membranes, the peptidoglycan, the attachment. Okay, so cells are growing happily in LB. Then you see that you have the small blue circles are the water molecules, and the bigger circles represent the solutes that are present in the cytoplasm, in the periplasm, in the environment. Then, paf, there is an hypoosmotic shock. Okay, so suddenly there is a drop in the concentration of the osmolytes in the environment, so more water. Okay, and then what? So water is then going to enter, more water molecules are going to enter the cell, and so the pressure will increase a little bit in the periplasm. Okay, this goes very fast, and then what? No, I need to introduce a new protein. Okay, it's just, I need to introduce it now to explain the model. And this protein is this one, the aquaporins. And someone mentioned the aquaporins before. So the aquaporins, so in E. coli encodes 1 aquaporin, AQPZ. It's not very abundant. It's like 80 copies, compared to LPP, 1 million, it's nothing. But it plays a major role. So it will allow water to quickly cross the inner membrane. Otherwise, you can delete the aquaporins. Water will be able to diffuse through the inner membrane, but it will be a slow process. If the aquaporins are there, then water will flow, through the inner membrane. Okay, so back to the model. So we have the aquaporins, so water will, enter the cytoplasm very fast, and this will lead to an increase in the turgor, in the cytoplasmic turgor, in the pressure, osmotic pressure that you have in the cytoplasm, because you have more water molecules coming. And this will, push, like I showed in the introduction, this will push on the inner membrane. So it will increase the tension in the inner membrane. Yes, with me, everybody? Okay. Again, I need to introduce another protein. And these other proteins with an S, they are the mechanosensitive channels. Okay? So E. coli encodes 5 of them, I think. And that's the structure of MSCS. That's one of the major ones. So basically, those proteins, they are present in the inner membrane. And it's kind of an emergency mechanism. When the pressure increases in the cytoplasm, then it will push, it will push on the membrane, so the tension will increase, and this will activate those channels, and they will open and release cytoplasmic content to the periplasm. Okay? And so water enters the cytoplasm, pressure increases. This increase in pressure will increase the tension in the inner membrane, and this will lead to the opening of the mechanosensitive channels. Okay, so to this point, everything I've told you now, as we are building the model, I mean, as it has been shown before, it's known, okay? And but no, I mean, things are going to be different. No, it's the new stuff. So now we have to consider the wild type and the mutant. Okay, so the MSCs open, they release cytoplasmic content to the periplasm, and then what? So when cytoplasmic content is delivered to the periplasm, this will attract more water molecules to the periplasm, in fact. Nobody thought of that before. So it will attract more water molecules. And so more water will enter the periplasm, and this will do what? This will increase the periplasmic turgor. Here we are in the wild type. So that means that the outer membrane is attached to the peptidoglycan. And so you will increase turgor, but there is a limited size to the compartment, okay? And so turgor will increase, and then at some point, it will counterbalance the pressure you have in the cytoplasm. And this will lead to the closing of the MSCs. And the wild type cells, they do survive. Now, when we look at the mutant, okay, the beginning is the same. So water molecules, they cross the inner membrane via the aquaporins, they enter the cytoplasm, okay? Tension increases in the membrane, the MSC is open, they release contents, solute, small proteins to the periplasm. There is these new, those cytoplasmic solutes, after being released in the periplasm, they will attract more water in the periplasm, in the cell envelope, but now the situation is different because the outer membrane is not attached to the peptidoglycan. So that means that if you increase turgor there, it will just push and push and push on the outer membrane. And therefore, okay, we have the bleb formation, you remember? Okay, so there is bleb formation, as you see here. So periplasmic turgor does not increase because instead of increasing turgor, you increase the volume because it's not attached. So periplasmic turgor does not increase. The MSCs remain open and cells die. And in fact, we think that they die because then the MSCs remain open, the inner membrane is not protected because there is not this counterbalance. That the cytoplasmic turgor is not counterbalanced by the periplasmic turgor. So the inner membrane, where you have like the electron transport chain, that we heard a lot about in the last 2 days, and the quinones and so on. So the inner membrane will break and cells die. And to support that, for instance, we can go back to the movies I showed you, and we can see that, okay, this is the inner membrane signal. If you look carefully here, you see that something is going on. We don't really know what's going on there, but we can see that the signal in the inner membrane, is impacted in the mutant, not in the wild type. And we see that the transfer of M-cherry, which is in the cytoplasm, to the periplasm occurs at the same time. M-cherry is too big to go through the MSCs. So for M-cherry to go to the periplasm, it must, pass through breaks in the inner membrane. Okay, so it's nice model because it changes the view people have on how bacteria deal with osmotic stress, on the role of the peptidoglycan. But when you have a model like that which challenges a little bit the view, then you also need to to obtain further evidence or further data supporting it. And so we have a few, and I will introduce you to 2 of them this morning. So what's really key, in fact, it's the buildup of periplasmic pressure. Okay, so in our model, this is what's really key. You need to be able to build up periplasmic pressure, otherwise cells die. That means that if you make the outer membrane stronger, then it should help. It should rescue a little bit, because if the outer membrane is stronger, then it should be able to withstand, more pressure, the buildup of more pressure in the periplasm. And how can you make the outer membrane stronger? You can do that by adding cations. Like in E. coli, LPS molecules, they are negatively charged. And so, you can bridge them using cations. And when you bridge them using cations, you prevent the repulsion, you make the outer membrane stronger. This has been shown, many times, so it's well established. And so here's the result. So this is we are looking at the LPP-OMP double mutant, with the single mutations in those two proteins. And this is, how they die when we expose them to an osmotic down shock. You see that when we add magnesium or calcium, so two cations that will bridge the LPS molecules, they survive much better. And if you look here, okay, with EDTA, of course, with EDTA, you're going to remove calcium and magnesium, and they die even faster. They become even more sensitive. One argument supporting our model. This meeting is being recorded. Oh, yeah. Okay. So I don't know why it's. Then we did something else. Remember the aquaporins? So the aquaporins, they allow water to flow very quickly across the inner membrane. So, but they can, AQPZ can be deleted. And then what happens is that just water will flow across the inner membrane, but slowly. And it changes everything, because when it's slow, then cells can adapt. They can change the production of osmolytes and this kind of thing. And so we said, okay, what's the impact of deleting AQPZ? It should help, it should rescue, because then water will enter the cytoplasm. There will be an increase in cytoplasmic turgor, but cells will be able to adapt. There might be some opening of the aquaporins, release of content in the periplasm, but again, they can adapt and so on and so on. And this is exactly what we found. And I got very excited when I heard, when Mike told me about that result, because this is how the double LPP-OMP mutant dies. And you see that when we delete AQPZ, you see that it rescues. And that's remarkable, because we can rescue the lack of connection between the outer membrane and the peptidoglycan. We can rescue that lack by deleting a protein, which is in another layer in the inner membrane, and that facilitates the entry of water. So, there was no obvious connection between those different processes, but there is one. So, and the other results, I don't have time to explain, but basically we use aquaporins mutant. They all confirm what I'm telling you, and yeah, and here above the green bar there, so we deleted aquaporin and we added magnesium, and then you see that there is even, a better rescue. Conclusions to this first part. Okay, so the first one is that the peptidoglycan is, of course, it's important. People have been studying the peptidoglycan for decades, and we are not saying that the peptidoglycan is not important. It is important, but its role, in fact, is different. It acts more as a scaffold than as a cell wall. It's a platform, and the outer membrane needs to be somehow attached to the peptidoglycan, in order to allow the buildup of periplasmic turgor. In fact, the key thing is that it's key to be able to build periplasmic turgor. And this is new. This will protect the inner membrane from lysing, from breaking when the cytoplasmic turgor increases too much. Okay, then the third conclusion is it's kind of linked to what I just said. So it's important to attach the outer membrane to the peptidoglycan. And so in E. coli, this attachment has been well documented. So, but in other bacteria, just a few years ago, it was not clear what was going on in other bacteria. But like one of my colleagues in Belgium, Xavier de Boll, found that in alpha proteobacteria, so not like E. coli, their beta-barrel proteins are also covalently attached to the peptidoglycan. So this idea that the outer membrane needs to, or is connected, is attached to the peptidoglycan, is and that is the case in many bacteria, if not all gram-negative, I think this starts to really make sense, and this is being discovered. And then the last one, it's kind of a question that was asked by Tom Silavi. He's one of the experts on the cell envelope. In a recent review, he asked the following question. He said, why is the outer membrane essential, in fact? And then people, usually when you ask that to people tell you, people who know the outer membrane well, they tell you, but it's essential because you have essential machineries like the BAM complex. I'm going to tell you about the BAM complex in a few minutes, or the LPT machinery. And those essential machineries are in the outer membrane, so that's why the outer membrane is essential. Yes, but those two essential machineries, they're essential because they build the outer membrane, And so, okay, so the outer membrane is essential, and the only essential machineries that are there, they build the outer membrane. So why do we have an outer membrane? And so this might be the reason why. So it's when you have a thin peptidoglycan layer, so in gram positives, it's different. When you have a thin peptidoglycan layer, what's really key is to have the ability to build, again, periplasmic sugar. And the only way to do that is to have an outer membrane. And that's probably why, at least one of the reasons why the outer membrane is essential. Okay, so that's the end of the first story. And if you want, I can stop now for a couple minutes. And if you have questions, I already see one there. I will try to address them. Okay, I start with yeah. Yes. Yeah, so in fact, there are three connectors. So I didn't talk about the third one this morning, but when you delete all three of them, then cells are not even viable. Okay. And so you need at least some connection. And no, we don't really know. They probably play a different role. Maybe that where they bind to the peptidoglycan is different. The way they will be regulated is different. So that's something we're investigating. But you always need at least some connection. Begonia? Begonia. No, it's not a stupid. It's not a, so I'm repeating the question for the people online. So the question is, why do we see one bleb at one location, one position, and not everywhere? That's a good question. That's probably, I mean, the easiest way to interpret that is to think that it's probably a weakest, the weakest point in the outer membrane. Again, to go back to the question before, we don't really know where LPP and OMP are. So when we have the double mutant, we still have the third connector, and so which is PAL. And so maybe that this is a location where PAL is not present. And so that's why we will see that bulge there. But that's a good question. Marcelo? Okay. Okay.
So it might be ridiculous to the location of MSRC, but probably we have a patch that's over that side. I don't know. It's an hypothesis. The second question is regarding, I guess, the bacteria is remodeling the peptidoglycan along the culture of the peptidoglycan. You can check whether this phenomena also depends on the culture phase.
No. Yeah. So here, what I showed you today was, and like I kind of alluded to that when I introduced the AQPZ deletion mutant. So here we go very fast. So it's within a matter of a few seconds. So there is no way that the peptidoglycan could be modified. So it goes too fast, and that's also something that we verified, to see whether the integrity of the peptidoglycan when we delete LPP OMP or when we removed or when we prevent them from attaching to the peptidoglycan. So we verified that the peptidoglycan was not impacted. It's not the case. And so here in those experiments, we did them with growing cells, so in an exponential phase. Yes. I think we haven't done that yet, but I suspect, I predict it should be the same, in fact, huh? Because then, so as long as they're alive cells, then even if you're in stationary phase, if you drop the same mechanism should be at play. Okay, Christina. Yes. 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 We haven't done that. So you can play with the distance between the two membrane by making LPP longer, and or shorter, but then you activate a stress response too. But anyway, and it's true that this could have an impact, but we haven't looked at that yet. Yeah. Memo, and then oh. The distance. Yeah. Yeah, so it's a good question. So that's something we discuss, in the last paragraph of the paper. So obviously it must be different in gram positives because there you have a very thick peptidoglycan layer. But maybe that I think that is more like an intuition that this kind of mechanism is also going on in gram positives, because in gram positives you have kind of a periplasma, and people working on gram positives they're embarrassed, when they have to deal with that, but you have space between the cytoplasmic membrane and the thick cell wall in which you have like the DSB proteins there. So uh, if yeah, so that's why I think that this kind of mechanism is also probably playing a role there. Otherwise, you would have just the cytoplasmic membrane next to the cell wall, and then the cell wall would prevent the lysis. But it's not what I mean. It's the case. It's not what's happening. Memo. Yes. What about? Yes, that's a good question. Um, so when we knock out the MSCs, then cells, even the double mutant become even more sensitive. And so that's good. That fits with the model. Now it's a little bit so difficult to really build on that result because even when you delete just the MSCs and you don't touch LPP and OMP, they also become more sensitive. So it's like comparing in like two thick cells, and then okay, it goes in the right direction, but it's difficult to extract a definitive conclusion. Okay. Okay. They survive. Yeah, they will adapt. Yeah, they will express that they will change like in the periplasm. They can induce expression of osmoregulated glycans there. They will adapt in the cytoplasm. There must be similar mechanism going on. They will adapt. And in fact, even with the wild type, if we take the wild type if we subject the wild type to a huge drop in osmotic pressure, even the wild type, if it's sudden, even the wild type will die. So if you take the wild type very high and then drop it, then it will die. But if you do the same, step by step, it will survive. Even and the double mutant too. Adaptation. Yes. Okay, are you ready for the second story? Okay. So the second story, we stay in the cell envelope. Okay, it's different, very different. So now you remember maybe I had the same image on the last slide of the previous presentation. So this cell envelope, the two membranes, the peptidoglycan, it's proteins there, disulfide bond formation. It's about like 600-700 proteins, different proteins will be expressed in this periplasm and in the outer membrane. So it's a substantial part of the bacterial proteome. Okay, and this cell envelope, it's really key for bacterial growth and survival. It's not you know that because like I showed you that when we prevent the attachment of the outer membrane to peptidoglycan, they become more fragile. But if you make the outer membrane if you decrease the integrity of the outer membrane, they will become more sensitive to antibiotics, and so on and so on. So it's really key for gram negatives to build and assemble the cell envelope and the outer membrane, in the best possible manner. Okay, otherwise they're gonna suffer. And there are still many things, that we don't know regarding the assembly of the cell envelope, of the outer membrane, and also the protection and maintenance. And this is going to be like the background of the second part of my talk. So this second part also starts with Seung-Young. So in fact, Seung-Young came to my lab 2010, and back then my lab was only doing redox. So we were only studying like a redox activated chaperones, disulfide bond formation, and so on and so on. And so we had a side project on this protein, which you see here. You see the blue protein RCSF. And why did we start working on RCSF? It's because RCSF has two disulfide bonds. And so we said, okay, we're going to study, this protein, the oxidative folding pathway, and see whether the SBC is involved, the SBD, and so on and so on. So we had a paper in 2011 on RCSF, and so that was more or less the time Seung-Young came to my lab. And then I still remember the day when he came to my office and he said, look at that, so RCSF, this protein that we just, studied, we investigated the folding pathway of RCSF. I kind of knew it was a sensor, a stress sensor in the outer membrane, but we never cared about the function of RCSF. And so Seung-Young came to me and he said, look at that. And that's a review that was published in 2005 by Susan Gottesman, so it was like 5 years before our project on RCSF. And as you see, you see those lightning, so it makes stress, that means there is stress. And RCSF was known to be a stress sensor. But as you see, okay, question mark, question mark, question mark, nobody knew how this protein could feel stress. And then Seung-Young said, don't you think that it would be cool to start investigating this protein? And I immediately said, oh yeah, that's in fact, it's a cool question. It's different. At some point, sometimes you get tired of project and you need something new. And so that was a new challenge. How does RCSF do to sense stress, to feel stress? Completely different. And so now we are in 2023 and we are still, trying to understand how does RCSF do to sense stress, but, it's like yesterday someone showed a puzzle. And so, now we after 13 years, we have some pieces of the puzzle, and I'm going to introduce you to those 5 pieces, and we're going to build a model together. Okay. So when you have this kind of question, how does RCSF feel stress? It's kind of a big question. I think everybody agrees that, okay, if you can understand how another membrane lipoprotein can feel stress and then send a signal to the control center in the cytoplasm. So we see that there is an intellectual challenge there. How does that work? But it's not easy to go from that big question to an experiment you can do. And so we did lots of things. And one experiment that worked after trying lots of many things was like a simple cross-linking experiment, simple pull down. Sometimes they do work, not always, but sometimes they work. And so what we found is that, when we cross-link RCSF using a chemical cross-linker, when we pull down RCSF, so that's the first piece of the puzzle, then we can see that we have 2 major bands. Oops. So one here, and I will come back to that one later, and this one at the top of the gel. And so using mass spec, using antibodies, we could easily identify that band as being BAMI. And when I heard that RCSF was interacting with BAME. Again, I got very interested and very excited. Why? Because BAME, it's one of the only two essential proteins in our complex. BAM is one of the only two essential complex in the outer membrane. There are not that many, LPT and BAM. And so finding your favorite protein interacting with an essential complex in the outer membrane, a complex, you know, that many people are caring about, also because it's a good target for new antibiotics and so on and so on, then you think, oh, I'm lucky, you know, because you could have found like another Y protein that nobody knows what it does, you know, but here we got BAME. So let me tell you a little bit about BAME. So BAME has a beta barrel domain, and as you see, a periplasmic extension of 5 tetradomains. So BAME is a beta barrel, and what it does is to introduce beta barrel proteins in the outer membrane. So it's the factory, okay, that makes beta barrel proteins. And in our mitochondria, we have a homologue of BAME. BAME doesn't do the work alone because BAME will collaborate with lipoproteins like LPP, I told you before. And there are 4 in E. coli, there are 4 lipoproteins, BAM-B, and then BAM-C, D, and E. And as you can see, they are in the periplasm, they're attached in the outer membrane, and they bind to the tetradomains. And so that's really the factory, the machinery that will introduce, that will fold beta barrel proteins in the outer membrane with homologues in chloroplasts and mitochondria. BAME was discovered in 2005, so not that long ago, in fact. So BAME, that's another thing you need to keep in mind, is that BAME comes in 2 different flavors. You know, if you look at the structure here, okay, you have the structure on the left, where you see a lateral gate with the 2 beta strands in yellow, which is closed. But then those 2 beta strands, they can undergo a scissor-like movement, poof, and they open. And that's the structure on the right. So it will open a gate to the outer membrane. And you see that the tetradomains in the periplasm, you know, will be impacted by that. Okay? And it's very likely, even though people are still struggling to understand, you know, all the details of the mechanism of BAME, but it's very likely that this opening, closing of the lateral gate is key in the function of this protein. Okay, so first piece, you know, and then I will have a small animation to put everything together. BAME, the protein that assembles beta barrel proteins, interact with RCSF. Second thing, okay, look at that. Second thing is that this interaction between BAME and RCSF is related to stress sensing. Why? Because we cannot delete BAME, it's essential, but we can deplete BAME. Oops. And so when we deplete BAME, then, and if we look at the activation of the stress system that is under RCSF control, we see that it induces that stress system. So that means that this BAME-RCSF interaction that we just identified, it's related to the stress sensing activity of RCSF. And I have further evidence. Okay, here what we did in those 3 different experiments, and you can simply look at the one on the right. Okay, in these 3 experiments, what we did was to activate the stress system by using antibiotics, polymyxin, for instance, which is colistin, or mesalinam, and so on and so on. And you see that when we activate the stress system, this BAME-RCSF interaction is gone. Okay, that's the second piece in my puzzle. BAME does interact with RCSF, one, and the second one, this interaction is related to the stress sensing mechanism. Okay, third one. We not only have BAME interacting with RCSF, but look, this is an old friend. You remember OMPE? Okay, so RCSF does also interact with OMPE. And not only with OMPE, but also with OMC and OMPF. And so OMPE, C, and F, okay, they are the 3 major beta barrel proteins that you find in the outer membrane. They are structurally different. I don't have time to enter into that, you know, but for my talk today, you can just remember that RCSF does interact with those OMs. Okay, OMC and OMPF are porins, OMPE is not, but whatever. Okay, so the 4th piece, so we have 5 in total. Okay, so we have seen 3. This is the 4th one. We have evidence that suggests that, in fact, the 2 complexes that we identified, BAME-RCSF and OMPE-CF-RCSF, they are not equivalent. One of them is catalytically active. One of them, in fact, is kind of an intermediate, and it's the BAME RCSF complex. So we think that BAME will, in fact, funnel RCSF to the beta barrels. Remember, BAME folds beta barrel proteins. And so the idea is that BAME interacts with RCSF. You have a beta barrel protein coming, and BAME will assemble the OMP-CF RCSF complexes. That's the fourth piece. And like evidence for that, for instance, you know, when we deplete BAME here, we lose both complexes. But when we delete OMP, it doesn't have any impact on the BAME RCSF complex. So that's another argument to say that BAME RCSF forms before OMP RCSF. Another argument is here. Remember that BAME functions with BAM B, C, D, and E. So if you only have BAME, it's not functioning. So here in this experiment, what we did was to overexpress BAME alone. So you overexpress a non-functional version of BAME. And when you do that, you know, you see that then BAME, you know, becomes very egoistic and keeps RCSF. It doesn't want to funnel RCSF because it cannot, it's not functional. And you lose those complexes. Okay, that's the fourth piece. And then the last one is that, and that's something which also came as a surprise a few years ago. So we found, in fact, that RCSF is most likely on the cell surface. Why did it come as a surprise? Because like a few years ago, the dogma was that lipoproteins in E. coli, it's not the case in Borrelia, in Neisseria, but in E. coli, in bacteria like E. coli, you know, the model was that lipoproteins face the inside. So it was not expected to find any lipoprotein on the cell surface. And we found RCSF on the cell surface, and we have evidence suggesting that the OMS play a role in that exposure. But no, there is still a big controversy, I mean, big, you know, to the level of our research and to our lab. Like another lab is saying, oh, this is how it goes. We have arguments against that model. We think it's that way. So that's a simple question, but, you know, we have been working on that question for years and years and years, because it's more complicated than it seems to really address the topology of that complex. So now we have those 5 pieces and we can build the model. In the absence of stress, so we think that RCSF will be transported to the outer membrane, will interact with BAME, and BAME will assemble the complexes between RCSF and the OMS. And RCSF will be on the cell surface, whatever the topology, you know, but it will be stuck in those complexes with OMS. And so this is the downstream component of the of the stress signaling cascade. Okay, that's something that we identified also. And so when RCSF contacts that green protein, that means the bacteria know that there is a problem. Okay, they need to respond. And so under stress, we think that what's going on is that RCSF, you know, will not interact with BAME. Therefore, it will not be exported on the cell surface. It will remain in the periplasm. And when it remains in the periplasm, then it can reach the green protein in the inner membrane and activate the system. You know, tell the control center, wait, we have a problem. You need to do something. Okay? I still have a few minutes? Okay. So that's the model. But again, when you have this kind of model, you need, I mean, we already, I showed you some experimental evidence supporting that model, but we needed more, and we are still working on that, you know, and it will probably take a few more years, you know, until we have a clear, solid understanding of what's going on. But so we were very puzzled by this interaction between RCSF and BAM, and so we wanted to know, okay, but how does RCSF bind to BAM? Remember, BAM has those 4 lipoproteins there. It funnels beta-barrel proteins in the outer membrane. I mean, how does RCSF interact with BAM? First, and second, what would impact the interaction between RCSF and BAM under stress? And so we decided, so Begonia told us yesterday about like, crystal structures, and so we decided to go after the crystal structure of the BAM RCSF complex. And this is something we did in the lab in collaboration with Han Remote in Brussels. And so this is the structure that we got of the BAM RCSF complex. We were very surprised when we got that, because we convinced that we would find RCSF binding to one of the potradomains there. That was our expectation. That was the most likely. We did not expect to find RCSF inside the barrel of BAME, but in fact, it is inside. And that's something that we could confirm using, I don't know if you are familiar with that technique, but it's very powerful. It's a site-specific photocross-linking. And so you can introduce a photoactivable residue at the position you want, okay? And so you do that in vivo, so cells grow, they make the protein with the photoactivable amino acid, and then at some point you expose the cells, living cells, you expose them to UV, the photoactivable amino acid gets excited and it binds to anything which is in close proximity, okay? And so what we did was to replace some of the residues inside the barrel of BAME by diazepike, which is one of the two cross-linkable amino acids that is available. And, we could see the formation of UV-dependent complexes here between BAME and RCSF when those residues facing the inside of the barrel, were replaced by diazepike, which means that, that confirms the structure, I just showed you. Okay, so now, it's nice to have a structure, but what can we learn? What does it tell us about the function, about the mechanism? So it was very striking to see that RCSF binds to the gate of BAME. You remember? BAME can open and close. Okay, so RCSF was found binding to the gate, ready to go out somehow. And this is something which is shown here. And in fact, if you try to see whether this binding is compatible with the open conformation of BAME, no, it's not. When BAME opens, RCSF cannot stay there because most of the interaction interface, is really in the gate area, in the region that will open when BAME functions. Okay, I will skip that just to confirm that. And then another, so first thing, the first information that we obtained, extracted from the structure, the second one is here. We saw that in the structure, we have this, the P5 potradomain, so the fifth potradomain is displaced by 20 angstrom. And then when we put that together, and so yesterday we heard about molecular dynamics, and so we use molecular dynamics to see, okay, what's going on when we go from BAME in the closed conformation to BAME in the open conformation, when we have this, potradomain number 5 moving back, underneath the beta barrel, when RCSF is inside, what does molecular dynamics tell us? And so this is the, if it wants to, molecular dynamics doesn't want to tell us anything today. Come on. Okay. Can I go back? Yes. You see? So when you open BAME and you bring the potradomain number 5, back under the beta barrel, you see that it pushes RCSF. In fact, we stopped because when the guy in my lab doing the molecular dynamics, came back, you can go on, and then you really see RCSF leaving the cell. And I told him, okay, we'll stop there because then, I don't want to use this as an argument for the topology. It does suggest that RCSF will go with the globular domain on site, but we have to be careful, And so I said, okay, we stop there. I want it to be an argument for the assembly, and so I'm almost done. So this is the model. So the current model on which we are still working, because now like for instance, we would like to have the structure of the full BAM with RCSF, not only BAME and RCSF, but all the lipoproteins there, which is being very challenging. We are using cryo-EM for that, and we are doing lots of other things. So we think that RCSF will remain, okay, with BAME, close BAME, then you have a beta barrel protein coming, and so, okay, and in the absence of stress, then, okay, BAME will funnel RCSF to the beta barrel protein to assemble the beta barrel RCSF complex. This might be a general machinery for the export of lipoprotein to the cell surface. That's also something that we are investigating to see whether it's general or not. And like I said, when there is stress in the cell envelope, then this will, I don't know why, it's getting tired. Okay, so it's like a, it's like a tracker. So we think that RCSF, and you think about that, it makes sense because it's crucial for bacteria to make sure that the cell envelope is well assembled. So one of the best way to see whether the cell envelope is protected, is well, is to monitor one of the two essential machineries that is present in the cell envelope. Because obviously, if you have a problem with like the BAM machinery, for instance, this will impact the integrity of the cell envelope. And so it makes sense to monitor BAM activity. So RCSF is really like we think it's a tracker, that will monitor BAM's activity, make sure that BAM functions well, and so on. So there are still many questions. So that's also fascinating because, E. coli, I mean, it's been studied for 138 years. So it's a very long time. And like with all those questions, we still don't know what's the topology of the beta-barrel lipoprotein complexes. How many lipoproteins are there on the surface? Does BAM serve as a general machinery? Are there other export machinery? And then this is really the last slide. That's something we published a couple of years ago. And again, that was amazing to me to think about that. So we looked at the sequence of the 80 lipoproteins that are encoded by the E. coli genome. And we found that half of them, they have an intrinsically disordered region at the N-terminus. And so why? Why it must have a function. So we started to play with those regions, and we showed that when you delete those regions, you really impact the targeting of the lipoproteins to the outer membrane. And again, so now we are still trying to elucidate the mechanism. And so that's the lab a year ago. So now some of those people are gone. Other people have joined the lab. And so, of course, I thank them because they do the work, they do the experiments. I thank the funding agencies. Thank you again for the invitation. Thank you for your attention. And if you have questions on this part, and if we still have time, I'll be happy to take them. Thank you. Memo? How it uses the strength. Transduce, yeah, so RCSF has a long linker like that, so the different potential mechanisms. So first, if BAME is not functioning well, so BAME will not export RCSF on the cell surface. So RCSF will be released to the periplasm. And from the outer membrane, it can reach IgA because it has a long linker, and that's something that we could show. And IgA has an amazing affinity for RCSF. It's like 1 nanomolar. And what's fascinating is that you only have like 300 copies of IgA and 3,000 copies of RCSF. So that means that you need a very efficient, with the high affinity of the green protein, IgA for RCSF, you need a super efficient mechanism to prevent IgA to stick to RCSF. You need 10% of RCSF molecules escaping that system to have a full activation of the RCS system, which you don't have. But to go back to your question, RCSF remains in the outer membrane and it can reach IgA. Another potential mechanism is that when you have you mess up the assembly of the cell envelope, then you can keep some RCSF molecules in the inner membrane, and then again that will activate the RCS system by allowing RCSF to reach the green protein. Diana? Yeah. So you. Yes. Yeah. Yes, it is a very good point. So I mean, RCSF is not a protein which is, I mean, the levels of RCSF are always constant. So it's not the production is not adapted. So it's not increased or not decreased. And you have, like I said, 3,000 copies of RCSF, and you have more or less 3,000 copies of BAME. So it's more or less 1 to 1. Now, BAME, when you have a stress in the other membrane, then you have a sigma E, which is a stress sensing system, which will induce the production of BAME. But if you have more BAME, it's fine in terms of RCS because it just keep RCSF away from IgA. And now in the lab, when we play with those levels, that's something we did. You know, when you increase the BAME levels, it will bring the RCS levels down. When you increase the RCSF levels, it will bring the RCS levels up. Because, like you said, you need to fine tune this. Yeah. Yeah. I mean, so NLPE is another lipoprotein that activates the CPX stress response. And there are 5 in E. coli. You have 5 stress responses active in the cell envelope. RCS and CPX are the 2 major ones with sigma E. And so there are similarities and differences. RCSF is really crucial for the functioning of RCS. So if you don't have RCSF, cells will be blind to stress impacting the other membrane and the peptidoglycan. So it's really key. NLPA in NLPE is less clear, even though we have published also on that, but it's been published that it might detect surface attachment, which has been then disputed. It might monitor NLPE, the lipoprotein transport, because when it's stuck in the inner membrane, then it can reach CPXA and activate the CPX system. It might also monitor the redox state of the cell envelope. That's something that we publish because when you don't have a full oxidation, you know, then it will activate. And but CPX will function really well without an LPE. So it seems to be like for lipoprotein transport defect, for instance, it will be important, but it's less well clear. It's not well established with an LPE. And then to go back to the disulfide question, when you don't have this disulfide bond forming in an LPE, it will activate CPX. When you don't have disulfide bond in RCSF, RCSF will be degraded. So it will have no impact on RCS. Yeah. No, that's something I forgot to mention that at the end. So we think, but that's something we are investigating as we speak, as I speak. We think that, and it makes sense in our model, that when you decrease the integrity of the other membrane, for instance, by changing the homeostasis of the lipids, you know, less LPS or whatever, that it will favor, it will, like the open conformation of BAME. And then if you favor the open conformation, it will activate RCS. We also have some evidence suggesting that BAME might be degraded under certain conditions. So we think that BAME's activity is impacted by the state of the cell envelope. And in fact, so this is not something from our lab. We have been just collaborators on that project, but like one of my colleagues in Brussels, he has a very nice paper coming out on a slide on SLIB, which it's another lipoprotein, and it's probably the first example of an intra-membrane chaperone. So when you so what he found is that when you destabilize the other membrane, like for instance with EDTA, you remove the cations, then the LPS leaflet will be impacted, you know, and the other membrane proteome will be destabilized. And so what SLIB does is to jump, if I can say, inside the membrane, and it will form like a rings around some of the beta-barrel proteins, protecting them from stress, you know, and I don't know why I said that. Anyway, it's coming out, and I think it's very nice story because it's the first example of a protein that functions as a chaperone inside the membrane. Yes. Yes. Okay. So first question. So does RCSF impact the activity of BAME? So that's something that we discussed in the lab just a few weeks ago. So we have no evidence that it does. So it's been reported that when you delete BAM, it was another lab. When you delete BAM B and BAM E for sure, and then I don't remember if it's BAM B or C, whatever. When you delete those two of the BAM lipoproteins, then cells die. But you can rescue cells by deleting RCSF. So probably that then when BAM is not functioning well because you deleted some of the lipoproteins, you remove some of the lipoproteins, then it can be toxic to have RCSF binding there. Yes, but I think that when cells are growing well, I don't think so. I think RCSF will transiently interact, and as long as the complex breathes and opens and closes, I have no reason to believe that it can be toxic. Second question, the function of RCSF outside. Maybe. Yes. So we don't know. So that's something that when we found RCSF on the surface, one of the first questions we asked was, what's the function? Is it just a mechanism to make sure that to export RCSF to make sure that it does not interact with IgA, or is the function when it is there? So at some point, Young did some experiment to see whether there was some cell-cell communication and this kind of thing. We didn't find anything. That doesn't mean that it doesn't have a function. It may have a function. It may not. If it doesn't have a function, then you may think, wow, that's a costly process because you export. At the same time, maybe the price to pay to have a very sensitive mechanism. I don't know. Yeah, but we could mutate. So the question was, could we mutate some residues that would not impact the interaction with BAME and everything? But the RCSF is small, and so the risk is that you might also impact the interaction with IgA, the green protein. But let's say that we find some mutations that do not touch the interaction with IgA, BAME, and what we know. Yeah, maybe, but we still, what would you test? Yes, yes, true. So that's we we are still stuck, with that first question because no, we have an idea of what's going on in terms of mechanism, but still, no, we feel that we have the duty to really have a solid mechanism. And thank you.
Thank you, Jean-François, for both talks. Very, very nice presentation. Hopefully, the first of many here in Uruguay.
I hope so.
A break now, about 10 minutes, and so we switch gears to Frederick. There is coffee break outside this room. No, I'm going. Okay. This is a card.
Okay, 1, 2, 3.
Bye.
Customer buys. Okay.
Hola, hola, hola. Okay, now I stay in English. Thank you very much for all you can hear. The last season of this workshop, at this time, the second of the day here at Pasteur Institute, we give from Frédéric Barras. Frédéric is professor at the Université at Paris, and also the director of the Department of Microbiology at Institut Pasteur Paris, and the head of the lab that is called Stress Adaptation and Metabolism in Bacteria. So, first time was in Uruguay. Thank you very much. And it's a pleasure to have you. You are part of the Pasteur family, so welcome to Uruguay.
Thank you very much. Is it okay with Mike? Do you hear? I don't know. Where is? Oh, he's gone. Okay. Okay. So no. Okay. Okay. So yeah, first time in Uruguay, first time in South America, and really a great first time. I really loved those two days we had, and thank you again for the invitation and for your enthusiasm and your contribution, and thank you for the meet. No, something like that. Okay, so what I'm going to talk about is iron sulfur cluster based biology, because usually when I talk is to geneticists, to biologists, and iron sulfur cluster sounds so much as chemist thing that you have to reassure them right from start. So we're going to talk about biology, okay? And so and of course redox adaptation. So we've been working on the iron sulfur cluster since about 20 years now, if it's not more. And we've been looking at various aspects of it, the biogenesis. I will talk a bit about that, the role they have in certain proteins. I won't talk about that. We are currently working on a tRNA modifying enzyme that is using iron sulfur. What we did recently, it's the history and evolution of the machineries that are important for making iron sulfur cluster, and this is what I will talk about mostly at the end of my talk. Okay, well, thank you. Any question? Okay, so because I have to tell him that I understood what he's asking. Okay, so iron sulfur cluster and iron sulfur proteins, they are all everywhere. So of course, this crowd knows very well what iron sulfur clusters are. Just as a reminding, this is the 4 sulfur atoms which are connected to 4 iron atoms in a so-called cubane type of iron sulfur cluster, which is 4 iron, 4 sulfur clusters. There are 2 iron, 2 sulfur, there are 3 iron, 4 sulfur, and so on and so on. But this is the most distributed and mostly frequent found in biology. So those guys are cofactors of proteins, and as you see, as illustrated here, all all living organisms on Earth are relying on iron sulfur-based biology, beside lactobacillus and I guess a few intracellular pathogens. So what I'm going to talk about, and here's the outline of my talk, what iron sulfur protein is good for, how to make them in vivo, how to keep making them under all conditions, all fluctuating conditions that Jean-François referred to as stress conditions and changing conditions. And I will end up with the history and evolution of the machineries. So what are they good for? So in the 60s, they were known essentially to be important for electron transfer in respiratory chains. Here you have a complex one, and you see the series of clusters here, which are throughout 7 or 8 proteins, and those guys are really allowing electron to flow. So just like a electric wire, okay? In the 70s, enzymes relying on iron sulfur clusters, but not on the redox aspect of it, were discovered, in particular aconitase. In this case, the cluster is the catalytic site. He has an iron which is sticking out of the cluster and which is acting as a Lewis acid reaction and transforming citrate in isocitrate. And in the 80s, a series of discoveries showed that the series of work showed
iron sulfur are also used by a transcriptional regulator because, of course, iron sulfur allow protein to sense change in redox, and redox being oxygen, NO, superoxide, and so on and so on. Okay, so this is the history. Now I'm going to tell you a bit, give you a few examples of them, which will show you how broad and how wide the effect of the use of cluster can be found in biology. So here you have one of the most subtle type, and I hinted about in one of the talk yesterday, I think, or 2 days ago. This is SOX-R. SOX-R is a transcriptional regulator which binds DNA and activate transcription of one gene, SOX-S, which downstream has a full regulant to it. And so, and that guy is working with an iron sulfur cluster, which is 2 iron 2 sulfur clusters here. And you see, in this situation, it is not active. Okay, and the reason it is not active is because you see the -35 and -10 are on opposite face of the DNA, and therefore transcription cannot start. But upon oxidation of the cluster, which becomes 3+, there is a change in conformation of SOX-R, which is somehow compensating the distance, which is a suboptimal distance between the -10 and -35, and that allows to fire transcription. This is a model which has been a textbook model, which has been in the literature since 20, 30 years, Hidalgo Temple thing. And this, I'm not sure we understood it all, because it seems quite okay. In any case, you see here, having the cluster or not doesn't matter. The protein is always on the DNA. What matters is the redox state of the cluster. Here we have ISC-R, another regulator. We'll talk about, we'll use that regulator a lot downstream in the talk. So that guy is, this regulator is the iron sulfur homeostasis sensing regulator, ISCR. In this case, it exists under two forms. One which has a cluster, which is here, and as such, it can repress transcription of certain genes, including ISC. We'll come back on that later. Upon conversion from the hollow form, that is the one which has the cluster, to the apo form, that is the one which lost the cluster, and lost the cluster because it was, for instance, destabilized by ROS, destabilized or not built enough efficiently because of lack of iron, whatever the condition. It turns into an apoform, but that apoform has also an activating, a repression, transcriptional activity, which is dedicated to activate the expression of other genes. So you see, in this case, you have two forms, one with the hollow and one with the cluster and one without the cluster, but the two forms are both active on different set of genes. Now, the most famous case and the most spectacular case is the aconitase, because that guy, that protein, of course, you know, aconitase is an enzyme. I just mentioned it already. It's working with an 4 iron 4 sulfur cluster, and as an enzyme, is converting citrate and isocitrate. Okay, fine. When it loses the cluster, it becomes an apoform, and that apoform is actually capable of binding RNA secondary structure, which are upstream regions, upstream genes that are involved in high iron homeostasis. Okay, so here in this case, you see you have a hollow form, which is an enzyme, and a apoform, which is a transcriptional regulator. So those three examples give you the illustration of how important iron sulfur cluster can be for the function of protein, and how rich they are, and the enrichment in terms of function they can provide to the cell, because you can make an enzyme becoming a regulator, you can, and so on and so on. And so now in E. coli, we have recently made a new list, a new catalog of the enzymes which are proteins which are depending upon clusters for functioning. And you have about 181 proteins, and you see that most of them, so the color coded, that is respiration. So most of the respiratory enzymes, of course, are depending upon clusters. Okay, but what you see here is that, for instance, those guys, just taken random, this is gene expression. So all those guys are involved, so one way or the other, with transcriptional regulation. This guy here, the orange one here, is metabolism here. Here in the red one, those guys are involved, those proteins are involved in tRNA modification, and so on and so on. Bottom line, there is not a single process in the cellular, in the E. coli cell, that does not depend upon at least one iron sulfur protein. That tells you the importance. And again, the use of cluster varies from one protein to the other. Okay, so no question, it's fine. You knew that all, it's no problem. Okay, so how to make them now? So how to make them? Well, in the 50s, in the 60s, in the 70s, the answer was no. And this question was not a question, was not one, because it was extremely easy to make a cluster, an iron sulfur protein. You take the protein, put some sulfur, some DTT, and some iron, wait a few hours, come back, and you have your protein with a cluster. In vitro, there is no problem of maturating iron sulfur protein. And it's only at the beginning of the 90s that Dennis Dean was working on nitrogenase in rhizobium, in azotobacter, sorry, that he somehow thought that it was not sufficient and that spontaneous insertion and building of cluster could not arise in vivo condition, a recurring theme when you talk about protein folding and protein activation. And so he looked for genes that would not affect directly the nitrogenase structural gene, but would be important for the nitrogenase activity, and he came up with the so-called NIF genes. And the NIF genes were the first gene to describe the NIF maturation process, which is the insertion of a cofactor, which is including iron sulfur, but not only in the nitrogenase. It turned out that the NIF biogenesis system was the first system to tell us that there is a machineries here to be taken into account if you want to understand the maturation. Yet that NIF system was dedicated specifically to the maturation of nitrogenase and not about the other iron sulfur. However, making the NIF mutant was not killing fully 100% of the nitrogenase activity. 10% were remained, and that gave the idea that maybe there is a secondary machinery or some backup system that might be important. And this is why how it was the iron sulfur biogenesis system or systems and philosophy and strategy were discovered. 10 years later, people working in medicine, people sequencing genomes, people working in biotechnology, people working in pathogens, that were our case, all of us suddenly faced those genes that we did not understand the damn thing of what they were doing. We didn't even know what iron sulfur cluster was, okay? Yet we were all getting mutation, chunk of gene, disease related, associated with these genes. All of them were looking the same. They were present in eukaryotes and prokaryotes, highly conserved. And that is where really the iron sulfur biogenesis domain field started. That was end of the 90s. And 10 years, sorry, the time is flying. 30 years later, this is where we are sitting. So basically, we can differentiate two steps, the iron sulfur assembly step and the iron sulfur delivery step. The cysteine is the source of sulfur thanks to enzymes which are called cysteine desulfurase here, which are stripping the sulfur from the lateral chain of L-cysteine under the persulfide form, and is going to meet with iron, the origin and the source of which is still unknown, if there is a dedicated one. Both elements, sulfur and iron, meets on a scaffold protein where the cluster is formed the way it will eventually be found in the final apotarget. And in between that assembly here and the target, there are a series of genes, of proteins that are helping the cluster to go from there to there, and those is the transfer delivery steps. So this is the basic concept of iron sulfur biogenesis. Then depending upon the organism, you have, of course, a different number of proteins that are intervening at each of these steps. So this is E. coli. E. coli has two biogenesis systems, the so-called ISC1 and the SUF1. Those two systems are synthesized from two operons, the ISC operon and the SUF operon. The two systems share common homologous system or homologous proteins, like the cysteine desulfurase here in the ISC-S, or the SUF-S cysteine desulfurase, although this guy is a heterodimer. I don't go into the detail of which. The two machinery rely on carriers, which are orange code here, which is meaning that they are homologous in terms of sequence. But at the same time, they do not share at all the same strategy to build the cluster. So the core, where the factory, where the cluster is being made, are totally different. And they are totally different, but they do respect one rule, is the recruitment of generic system. I'm explaining that. So here, the chaperone, the scaffold is called ISQ, is where it is, the cluster is being made. Here, the scaffold is sub B. This is where the cluster is made. But you see those green guys here, this is DNA K, DNA J like, and these are ABC transporters like. And those systems, which are generic, were apparently recruited and dedicated to the making of cluster in the machineries, either the East one or the South one. So it's funny to think that the evolution used conversion somehow, because all of that is to make the same product by recruiting generic systems which are different. Yet they are all, both DNA, KDNJ, and ABC, of course, are using ATP. So probably this is the ATPase function, which is of use for the cluster here. Okay, so E. coli, two systems. One is working and expressed under stress condition. It's under the control of the fuel regulator, OXR regulator, ISR regulator, probably NSR regulator. So anytime there is a problem, stress problem, and stress, redox stress, or any other type of stress, E. coli will make the cluster using the SUF system. Anytime the growth, everything is fine and nice, it will be using the ISC system, which is under its own control, as I'm going to show you. So two systems, but before to go into the regulation, two systems, as I already said, that are really conserved throughout evolution. The ISC system is present in mitochondria, and the subsystem is present in chloroplast. And in eukaryote, you have a cytosolic machinery too, which is important for delivering cluster into the nucleus in particular. Here is just a hint to the importance of iron sulfur cluster in diseases. So this is this is the mitochondria, and this is the nucleus. Every time you have some mark here, it means here you go here and you have one of the terrible diseases it's causing. So I mean, there is a tremendous number of pathology which are associated with defects either in iron sulfur proteins or in iron sulfur biogenesis protein. So, fine, still with me? Okay, I'm with you. How to keep making clusters under all conditions, because of course those guys are essential. There's no such a E. coli, such a thing as an E. coli cell not making cluster. An E. coli cell not making cluster is a dead E. coli. I say that because it's been a great debate in the literature about the use of a strain lacking the capacity to make cluster. Those do not exist. They are not viable. Okay, so the problem with the cluster, and they have a bad reputation in the sense they are fragile. They are fragile. They are destabilized by oxidative stress, by nitrosative stress. So this is not really true. I mean, those who are really destabilized by that are the class of dehydratases. So this is about 10% of the iron sulfur proteins, and those are the proteins I already talked about with the aconitase that have a cluster which is fully exposed, surface exposed on the molecule, and therefore the iron needs to be exposed to the solvent, and therefore it can be also hit by a ROS or NOS, and those are the ones which are sensitive to oxidative stress. In any case, those enzymes, when they get hit, the cluster is destabilized, and a series of things ensues. The first one is that, of course, the enzyme loses the activity, so we have a problem here, but also the release of iron is going to fuel on the quantum chemistry, and you're going to produce additional ROS or a NOS, and so on. So this is a kind of bomb within, and this is the association between and the problem between oxygen and iron sulfur cluster biology. This is so well illustrated by a work by Italian colleagues who went to look at all the genomes they had in the data bank at the time, and what they looked at is it was the prediction of iron sulfur proteins per genomes. Okay, and so here you have one genome which is a Desulfovibrio vulgaris, which is a strictly anaerobe organism, and he dedicated about 4% of his genome to iron sulfur proteins. Here you have a good friend to desulfur Vibrio, Mixococcus, which is the same clade or the same taxonomy. I'm not a taxonomist, okay, but it's very related in terms of taxonomy, but strictly aerobic organism. And you see the number of iron sulfur proteins falls to 1%. Okay, so this is illustrating, and of course we have our E. coli, which is always a kind of center right, center left, and it is a kind of 3% average type of iron sulfur protein. So this is illustrating the deficit, the relationship between oxygen and iron sulfur. You need iron sulfur, yet you have a problem with oxygen. Did I went back? No. So back to the E. coli one and the regulation and the use by E. coli of those two systems. It's a kind of complicated slide I'm going to take you through step by step. This is the result of a lot of work of a lot of labs around the world, but mostly Trisha Kiley, Gigi Stoss, and Suro, who did, and Jim Imlay, who helped to set up this model. So what we're going to look at is what's going on in terms of iron sulfur production in E. coli strain growing. An E. coli strain growing under normal condition. In that condition, you have the ISC system, which is again synthesized from this operon here, which is going to make a cluster. And the cluster is going to go to one of those 181 proteins I showed you about, and among which one is a ISC-R. ISC-R being the product of the first gene. So that ISC-R gene is going to, protein is going to get the cluster produced by the ISC machinery, and as such is going to repress the expression of the ISC system. Repressing the synthesis of the ISC system, the ISC-R is going to switch to the apoform. Switching to the apoform, repression is going to be alleviated, and on and on and on. So we have here a rheostat-based type system that is ensuring that there is enough cluster in the cell for the cell to grow. Fine. So you have this equilibrium here, which is here from what I just said, depending upon the activity of the machinery, but also can be destabilized or perturbed by exogenous conditions, in particular ion limitation and oxidative stress. And as I told you at the beginning, the apoform of ISC-R not only does no longer repress, but it activates genes. And among the genes that it activates, the SUF, the second system. Now, if we are therefore taking back our E. coli, growing it in the stress condition, ISC-R is going to be mainly under this form, and as such is going to activate the subsystem. It's no longer going to repress the ISC system, so we will have an E. coli full of cluster, making cluster from the two systems, which is something which is not good, because first cluster are costly, and also those systems, if you think about, they are not that smart, because here, sub A, sub B, CD actually, those three guys or four guys are iron sulfur proteins per se. And here is U, is A, FDX are iron sulfur. So you are making a lot, but to make a lot, you are using a lot too. Okay, so this seems that there is an economy in the sense that you want to switch from one system to the other, but you don't want to keep the two together at the same time. And to do that, the FUR repressor, for instance, which is repressing the SUF under iron field when there's iron, but which is not going to repress it under iron limiting conditions. That is when ApoISC-R is also going to activate the system. FUR is repressing RIB, a small non-coding RNA. RIB is targeting the intergenic region between ISC-R and ISC-S, such that now when you are under iron limiting condition, SUF is expressed, but ISC is no longer expressed because RIB is going to catalyze the downstream, the degradation of the cognate small non-coding cognate RNA, messenger RNA region. Okay, and notice that it's acting here such that the ISC-R upstream gene that you need to activate SUF remains intact. Okay, so target of the small RNA is in between the structural and the regulatory, is an uncoupling of the operon structure. Okay, so this is beautiful. This is incredibly smart. This is really sophisticated. And what's for, I said, to guarantee a production of iron sulfur cluster throughout a wide panel of conditions. I give you an example now. we observed, and we and others long time ago observed that a mutant in the one of the ISC genes, ISC-S or ISQ-A, is fully resistant to gentamicin, which doesn't make any sense at first because gentamicin is an antibiotic and sulfur cluster. I mean, at the time there was no connection between the two. So of course we got into that story. We trying to understand what are the molecular connection between iron sulfur and antibiotic. And the molecular connection and the physiological connection is the following. You have here the proton motive force, which is required for aminoglycoside to be taken up by the cell and hit their target, which is the ribosome. And PMF is produced by the activity of the respiratory complexes, complex 1 and complex 2, which have plenty of iron sulfur cluster. So the idea was that depending upon how you make the cluster of the complexes, you might have different phenotypes, which was kind of weird, but okay. So we pursue that. And indeed, to make a very long story short, what we found is that when you have iron limitation, you use the subsystem, and you see the subsys is produced here. We have a growth, which is a culture which is grown in the presence of dipyridine, that is iron limitation. We add gentamicin, okay? So the antibiotic should kill E. coli, and you see that E. coli keeps growing, unless, and at the same time we look at the amount of subproteins, they are fully expressed here. Till the point we pour massive amount of iron in the culture, and immediately the cell becomes sensitive to gentamicin. The ISC start to, the SUF start to decline, and the ISC is emerging. So you see this regulation I told you between FUR, RIB, ApoISCAR actually is indeed allowing the switch from one system to the other. And the consequence of that are that, described here, the ISC system is extremely efficient in putting clusters into the complex 1 and 2. As such, the two complexes are making PMF and aminoglycosides are coming in. Now, when we switch to low iron condition, E. coli is using the SUF machinery, and the SUF machinery is good under iron limitation, but it's not as efficient as the ISC. Therefore, the level of the PMF produced is extremely low, sufficient to live, but not sufficient to allow the aminoglycoside, at least at this concentration, to be taken up by the cell. Okay, so you see that for going from one system to the other, you're still making cluster, but the way you make it are slightly different, and the phenotypic consequences are drastic. In one case, you die, in the other one, you survive. Okay, you still with me? I don't believe you. Yes? Okay. Now the history. How am I doing in terms of time? 15. Okay, so I told you that story about the connection and the difficile relationship between oxygen and iron sulfur, and this is something which has been part of our community. I'm a part of the community at midnight or 10 p.m. at the beer, at the counter, okay, just like talking about iron sulfur, nothing. And what we were saying in that was, well, iron sulfur has been used as soon as life appeared. Probably it was used also to shape and guide the evolution of life, okay? Nothing surprising. At the time, there was plenty of iron under the ferrous form, and there was plenty of sulfur, and there was no oxygen to put the mess in the system, okay? So this is the way life evolved and used iron sulfur and developed that iron sulfur-based biology. Fine. But it was so successful that at one day, I'm going to make it the kid way, one day, proteins with iron sulfur cluster became green, if I can say so. That is, photosynthesis emerged. And with photosynthesis, oxygen arose, and with oxygen, all the problems. One problem, the one with the dehydratase like, and also, but more importantly, probably, the oxidation of iron into this beautiful red thing, okay, which is absolutely not available for life. So the shortage in iron to make the iron sulfur cluster, that was not a good idea. And in addition, you had oxygen, which was produced by the photosynthesis. So we think, or we talked, and we said that maybe at that time, the idea, the temptation was to get rid of iron sulfur cluster-based biology and to replace it with a non-iron sulfur dependent biology. And actually, there were some key examples like fumarase and so on that could support such a view that more and more we are evolving toward getting rid of our dependence to iron sulfur. At the same time, as I said, today all living organisms on Earth have iron sulfur, are depending upon iron sulfur. So probably this was not the philosophy or the strategy that was used by the evolution, but rather, so what happened, that what we thought, maybe what happened is the emergence of the machineries, the SUF, the ISC, and the NIF machineries. So maybe those machineries that are used to make cluster and deliver it actually emerged as a consequence of the conditions that were really deleterious for iron sulfur-based biology. And we went on that. And that went on and went, as I said, in the bar. Nobody really tested it. And thanks to COVID, we had nothing to do but play with computer. And so I talked with my friend in Pasteur, Simonetta Gribaldo, who's a bioinformatician and phylogenomist, and we said, well, why don't we tackle that question once for all? And we did. So, so what they did, I mean, Pierre Garcia was the postdoc in between our two labs, and what we did was to go to, first he built up a non-redundant, clean database of 10,000 prokaryotic genomes. We don't care about eukaryotics in this field because eukaryotics got the cluster from the mitochondria from the rickettsia. So it's what we get for the prokaryotes is sufficient to understand the whole evolution. So 10,000 prokaryotic genomes, about 2,000-3,000 archaea and 7,000-8,000 bacteria. And we went after all, I mean, he went after all the genomes and looked for ISC-S, SUF-S, NIF-S, and trying to attribute, okay, this genome as ISC-S, this genome as SUF-S, and so on, which was not easy because those guys are sort of looking alike sometimes. So the criteria I used to decide who was what and what was who is here the series of criteria, the topology, the evolution, the alignment, of course, the length sequence, domain composition, and most importantly, the genomic context. Okay, so if you get a gene which is looking like an S, is it close from a gene which is looking like a U, which is ISC-S is U, or looking like a B, which is more like SUF-S, SUF-B, therefore that will be more a SUF than an ISC, and so on and so on. So doing that, he ended up with this type of thing. So I'm going to take this guy here. So this is the number of genomes here which we decided to have a SUF-S or CSDA, doesn't matter, it's another name, okay? So we had the kind of consistent set of genomes that have SUF-S. We have a series of genomes, we can definitely attribute them to conclude they are containing ISC-S. You know, these families were consistent according to the 8 criteria I showed before. But the thing which was strange is that we had one which was really, so we had NIF-S, YRVU, I don't go into those were known, but we have one big set of genomes that were containing something which was an S, but couldn't fit into the SUF-S, ISC-S, YRVU, or CSDA definition. So we said, maybe this is a new system in this SUF-S. It happened that those genomes that had that mis-S, the way we called it, actually when we looked at the U family, the U protein family, we found the SUF-U, we found the ISC-U, we found the NIF-U, and again, a set of genome had a U thing, but that was neither one of the known one, and moreover, those guys were the guys who had mis-S just adjacent to it. So we concluded that we had discovered a new biogenesis system, which is minimal, which is made of 2 genes, which we called mis-S and mis-U. And the same arose for the SUF system. So we had the genomes, we have SUF-B, genomes, we have SUF-D, and there was something which was sticking out here and was looking like SUF-B, yet was not SUF-B. And those guys who were in there were in this pocket too, that have something again looking like SUF-C, but not being SUF-C. So therefore, we conclude that we had a second unknown, a new family that we called SMS, for SMSC and SMSB. Okay, so that was the beginning of the discovery of the two new machineries. So what we did then was to look at who has what, okay? And so here is Ceph. Those guys are all the bacteria. Of course, you don't see, doesn't matter. You just, every time you have a dot, of course, it means you have one of the Ceph genes of A, B, C, D, S, E, and the size of the dot is the number of genomes in that category, in that class, or in that species that have it. Just going like that, I mean, we just, it's one thing which really jumped to us is that here are the genomes we have, which have Ceph, and here are the genomes which have ISC. So I remind you that ISC is the mitochondrial one, the one which is being used in, which is studied in humans. I remind you that ISC is the housekeeping one in E. coli. Okay, actually nobody has ISC, but the humans and E. coli. Most of the species have Ceph. The subsystem, which was a backup system in E. coli, actually happened to be the most distributed and the most used in nature. Then we have the SMS. SMS, very few bacteria have SMS, but interestingly enough, when they have Ceph, they don't have SMS, and when they have SMS, they don't have Ceph. So it seems that there is a kind of relation here. MIS was much more widespread, and NIF, as we knew it, was present only in nitrogen-fixing bacteria. That's all. Another irony of the story, I remind you that the whole field of iron sulfur biogenesis was discovered after discovery of the NIF system. Actually, it was just in one bacterium that those guys were existing. What about the archaea? The archaea, a lot of archaea have Ceph, confirming that Ceph is the most distributed system in nature. A lot of them have SMS, and again, we have this rule of mutual exclusion between SMS and Ceph. Here, those guys have no Ceph, but they have SMS. And MIS was kind of widespread as well. So another way to look at that here, the bacteria, most of them have Ceph, and when they have Ceph, they don't have MIS. When they have MIS, they don't have Ceph, so the overlap is here. And the archaea, there you can find Ceph, you can find SMS and MIS, and you see a lot of our archaea have both systems, SMS and MIS. So why is it so? This is the phylogenetic tree. Phylogenetic tree, I will just go fast just to show that when we look at the phylogeny of SMS or MIS, you have all the archaea here, all the bacteria here, and the same here. So this is monophyletic evolution. That means that those systems were present in the last universal common ancestor to bacteria and archaea, which is known as LUCA. And now that is back to our question. The question was again, is it true that Ceph, ISC, and NIF arose as a consequence of the oxidative stress due to aeration, oxygenation of the atmosphere? Well, if those guys were already present in LUCA, the answer is no. Okay, so the iron sulfur biogenesis systems have always been there, at least since LUCA. Just a detail here which is funny is that the cyanobacteria are totally out of the place they're supposed to be, probably because they acquired the Ceph system bilateral transfer. But this is just a detail. Now, are those genes encoding proteins? And are these proteins, I'm talking about the SMS, actually doing something with iron sulfur? Because this is just bioinformatics, okay? So we focus on SMSCB. I will tell you why in a few minutes. So we looked at their capacity to interact. This is a 2 hybrid. This is a pull-down experiment. C and B are there. Okay, so they form a complex as we would have expected. Do they carry a cluster? So we purify them and we looked at the UV visible spectrum, and we have the signature of an iron sulfur. So yes, they do bind an iron sulfur cluster. Now, are they able to transfer that cluster to a target? And this is what you have here. This is the aconitase activity, the APO form, no activity that we are mixing with SMSCD for 15, 30, and 40 or 130 minutes. And you see the activity is reappearing again. So indeed, SMSCB do bind the cluster and are capable of transferring the cluster to apoaquinitase in vitro. So this is clearly bona fide iron sulfur biogenesis system. Now, this is a structural model AlphaFold. I'm sorry, I don't understand much about bioinformatics, but even less to structural biology. So, so this, they are looking the same, okay? Well, not really. They are looking the same, but there is something weird that's SMSC here. This is SUF. SMSC here has 4 cysteines here, okay? You don't find any cysteine in SUF-C because in SUF, the cluster is in SUF-B, not in SUF-C. SUF-C is the ATPase. That is the one which we think is capable of promoting assembly, delivery, whatever, using ATPase, but it doesn't bind the cluster. In contrast, that SMSC, which is somehow looking like SUF-C, has this extension C-terminus that is looking like being able to host the cluster. Therefore, that SMSC would be capable of both promoting the transfer, but the assembly of the cluster, which is justifying that we call it SMSC and not SUF-C-like, like you can find in the literature. So this is the whole context of the SUF system. So that's a subsystem, SUF-B, SUF-D, SUF-C, SUF-C, a dimer, and with the ATP. They host the sulfur coming from the cysteine desulfurase and the iron, and make the cluster at the interface between B and D, deliver the cluster thanks to SUF-A, which come and goes over. Now look at the SMS, we have a big problem. We have no cysteine desulfurase which is associated with that. So where does the sulfur come from? First big question. SUF-C has again ATPase, but SMSC seems to have the cluster here. So is it true? Is it indeed a scaffold doing 2 things at the same time? So this is the questions we are currently working on. Preliminary evidences is that is indeed the case. So we have a new type of scaffold. More difficult is to find the source of sulfur. So there is roughly 10% of the genomes of the bugs which have SMSC, which grows under high H2S environment or hierarchies. So that has been already seen that there are organisms that have no cysteine desulfurase and for which it was proposed that they are just taking H2S from the outside. That will solve the issue for about 10% of the guys who have SMSCB. Remains a lot of them for which we need to find the sulfur source. And with this, I think this is the final scenario. So just to remind you that we identified in Luca 2 new machineries which are kind of minimal, MIS and SMS. So MIS was in some archaea kept as such. It was also acquired by the bacteria, probably undergoing a lot of duplication, loss, and so on and so on. It eventually became the SUF or became the ISC and the NIF here. We had a lot of lateral transfer here. I'm not going into the details. The thing which is important is that the cyanobacteria are here. That is where oxygen arose. And obviously, as I already said, the iron sulfur cluster to be made needed assistance from protein machineries well before the advent of oxygen, which in the context of those 2 days is really interesting and it's anecdotal because I happen to be old enough to remember that the guy, a young postdoc in Beckwith's lab, came up at the Cold Spring Harbor meeting and he was invited especially because he had found something that was weird. And I was shocked because he talked with a lot of anthropomorphism. And he came up and he was young and he said, if I were E. coli, I would not depend upon, like a redox condition and so for making iron sulfur, for making disulfide bonds. I would come up with protein. And indeed, is what I found. And that was Jim Bardwell. And that was a DSBA discovery. And from then on, it was obvious to me that, and that was back, that everything is catalyzed in life. So this is for the student. There is no such a spontaneous process, okay? And what we just found here, that just also for the sulfur biogenesis, Luca had to catalyze and to rely on machineries to make them, although there was no oxygen, plenty of iron, and plenty of sulfur. So I will leave you with a few, nature is a series of, we were talking about the other day with Marco, that it's a series of stamp collection more than models. And so this is what we have back to the ISK surf. So here you have all the bacteria, the gamma proteobacteria, and these black boxes indicate that those bacteria have ISK. And those black boxes here indicate that those bacteria have surf. Now, look at how many have both ISK and surf. Now you got the answer. 1, E. coli and Salmonella, maybe, okay? So this is just amazing. And so in a way, it gives some additional rationale to this incredible regulation, because indeed you don't want to have two systems at the same time, although they are essential, okay? And a funny one here is Pseudomonas. He's a really funny boy, this guy. Look at this, all those Pseudomonas have ISK. A few of them do not have ISK and they have surf. So of course it's essential. So they got it, but again, they didn't accumulate two. And I will finish up with the Shigella, which is spending its life to kill the surf system it has. So those are point mutation, deletion in dysentery, in Boydie, surf B, surf C are the targets, and so on and so on. So Shigella has two because in E. coli, but now it is specialized into a host. He spent his life killing the surf system, which is, by the way, surprising because surf is supposed to be the stress responding system and not the housekeeping one. But this is open for the future. Okay, so with this conclusion, I guess the conclusion is one of the interesting conclusion is this one, talks for models versus biodiversity, because obviously what we found is showing the limitation of model. And this is a group. Simonetta and Sandrine are in Grenoble and Pasteur were very important in this work. This is my group under stress condition here. It's osmotic pressure, in Marseille. And where is she? Emmanuel Bouveret, Pierre Garcia, the senior author of the paper, was a postdoc in between Simonetta and my group, did the whole work. Masha is a student who is taking over with Jean-Michel as well. Thank you for your attention, and thank you again. Thank you. Because, well, the E. coli view again of that was that surf is a stress responding system. Therefore, it was no surprise that surf was in the chloroplast because it's where there is photosynthesis, and therefore a lot of ROS being produced as a side effect. And therefore, it was making sense that the surf being more resistant to oxidative stress condition or environment was the one found in chloroplast. I'm talking about the days where we didn't know that surf is actually widespread. Okay, so maybe there is another one, but that is the reason, presumably. Biochemically wise, there are 2 or 3 papers that tend to support the idea that, for instance, ISK is more sensitive to a stress condition, that ISQ is more sensitive than SUF-BCD. So there separate elements seem indeed to be more sensitive to stress condition when you look at the ISK versus the SUF. But this is to be further demonstrated. Excellent question. A lot of work on that. So we can try to be synthetic. Not the dogma. Yeah, is that in E. coli, both ISK and SUF share the same targets. Okay. We, however, identified SOX-R as being matured by surf under under stress condition. Under non-stress condition, which doesn't make any sense because you use socks only under stress condition, you can mature socks with ISK as well. But very soon, ISK is getting inactivated by the stress, and therefore surf is taking over. So the answer would be there is no such a thing as substrate specificity. But what I showed you for the antibiotic thing is that the surf is not maturing as well the complex 1 and 2 than the ISK. So might be some differences. Of course, we are in biology. Okay. So and then we addressed that question in a paper that published 2 years ago. We took for biotech application. We took a series of ISP-G and a series of, can't remember, NAD-A, 2 enzymes with iron sulfur, and put them into E. coli and ask which one would be. And actually, it was really disappointing because the rule was that the further taxonomically wise it is, the least it is efficiently matured. But it was just a matter of taxonomy as for any gene you would express in an heterologous. So last answer, SMSBC, they have a cluster, right? That cluster, they got it in E. coli. So you see that an ISK system, which has never been an SMS, and even less than that, it's just mutually exclusive, still capable of maturating it. So there is no specificity. That would be my answer. Yes. No, so I got the answer since about 3 days. I asked Marie Touchon, she's working in with Denameux, Rocha, and she's the one who did the pan and the core genome of E. coli. So I asked her whether the ISK was present. So ISK is the core operon in all 2,500 E. coli isolates, or 2,000, I can't remember. In contrast, the SUP is fading away a bit, like in Shigella. 95% of the E. coli seem to start mutated, sub B, sub A, whatever. So they seem to be getting rid of it. Yeah, sure. I was exaggerating. There's Photorhabdus, which has 2, Salmonella, and E. coli. But no, but for instance, which one is, which is close from E. coli is another. A Vibrio has only Cytobacter. It has what? Surfenisk or only one? We can go back to it, but I mean, yeah, very few, and not fitting within, alpha, proto, beta, whatever.
Question, but going from the bottom always. What's the affinity of the ISK repressor for the iron sulfur class? Because you said when
affinity, I don't know.
But they need the or so no idea.
Oh, ID, sure. This is all the studies that Trisha Kiley is doing in Madison, but I don't have them in my head.
Regarding the provider of sulfur, this organism with the SMS, and do you know whether they have some sulfur transference? I mean, you don't need to, or yes,
I've been very interested by all the talks about H2S and sulfide and persulfide that has been during those 2 days. What we are trying to do is to use the E. coli approach again and to overproduce MST, which is a mercaptopyruvate, blah, 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 and to see if that could compensate for the lag. Well, I don't know. What do you think? Yeah, thank you. Yeah.
But of course, like these two have to be
Sure. No, we have no. Well, okay, too bad. That was the idea. I know we constructed the MST under the arabinose promoter, the PBA promoter, the chromosome, and we haven't seen any. But it's really hard to assay H2S as well. We do the strip, the paper strip, the kind of funny thing. Okay, good. So. Eukaryotes. Yes, the blastocystis and so on. No.
Let's shed light to whether a specific symbiont contributed to the system present in mammals. Do you think that in pigs at the very early time both existing and one was pulled out from our genome? So what's the—
No, but you think no, the surf was acquired from cyanobacteria. That's right. No doubt. No. Yeah. So we have a paper that just came out in PLOS Biology, which is on origin of the yeast and eukaryote. So I can't remember the name of the bug. It's a bit down further upstream of what it was thought to be. You know, it goes. I know there is a big debate upon the origin of mitochondria and so on, and it was not Rickettsier, but maybe one just before. And using the yeast and the surf, mainly the yeast in that case, Simonetta and Pierre supported the last version. I'm sorry, I don't remember the name of the class, but yeah, the paper just got out. So we use that as a tool. He's going to ask me bioinformatic things. Yeah. No. Those have like okay, so before we started, we looked at them as cysteine desulfurase for sulfur. They were known as aminotransferase class V and class whatever. And then it was two classes, class 1 and 2 for class 1 is yeast, class 2 is CEF and CSDA. Behind underlying or behind these classes, there are differences in biochemistry in KM, KKI, and so on. And structurally wise, there is a big difference in that the ISC-S have the cysteine, the catalytic site, which is a pyridoxal phosphate dependent enzyme. So all that is more or less exposed. You know, it's not very far from the surface. In contrast, the CEF-S, CSD, and so on have a catalytic site which is well buried inside. And in addition, it is transferring its sulfur to CEF-E, the small protein, which is accumulating per sulfur form up to 7, 8. So that's also where the idea comes from, that the CEF is for oxidative stress, because it would be operating under conditions where you don't want to mess around with sulfide and so on. So there are differences biochemically and structurally. Sequence-wise, it's 30% sequence identity or something like that, 25. Sure. Every time I've been asking, there are two questions, what you're saying. I mean, I've been interested in intellectually, because this is pure biochemistry and a very strong biochemistry, which is, do you acquire the cluster in a co-folding manner, or is the cluster a cork that wait for the protein to be folded and then you put it? Probably depending upon the protein, you're going to have that. This is for the last step. Okay. Then how do you transfer it? The only experiment that's been done is by my friend Marc Fontecave and Sandrine. It was on the maturation of biotin synthase. And it seems that there's a choreography between the different cysteines of the donor and the receptor. So you have the cluster is passing on from one cysteine to the other, and that's the way it is transferred. But one thing that we cannot get a target acting as a transferring protein. Once it's there, it stays. So it might have a lot of other criteria, stability, thermodynamically wise, and so on and so on. But we haven't, of course, looked at that. But yes, this is a black box for now. Okay, thank you very much for your attention.
Okay, thank you. Hello.
Hello, I can introduce myself. I'm Pascal Duran from Interactomics here. I have a very good collaboration with Anne-Marie Westerfeld.
Okay.
We're searching for pieces of cell division and molecular
Sure.
probably you. So it's really very nice. Very nice. We are here anytime you come.
Okay, I want to
Visit. Yeah.
Thank you. I have to sit down with you and explain about how it's done. Yeah. I don't know about how it's Yeah. So it's a very small only shop. That's no different than your yeah. So I'm thinking that
Let's go.
Yes. Can we leave
Just gonna close the door because if you want to start
Yes.
no problem.
No,
No problem, no problem.
I'm gonna ask them to close the door. Okay.